the condition of symbiosis brings, for instance, together what? The fact that after the infection thread make a change in the alignment of the cells of the plants, and there is the formation of nodules. That looks like, being a gastroenterologist, like the polyp of the intestine, for instance, of the colon. So, for instance, if we want to just move, excuse me, yeah, see, okay. If you just take a look at what these studies have brought, there is an evidence that the Rhizopia infection induces the differentiation of the host cell, which is at the basis of the nodule formation. That is important for this relationship for the plant. And if we look in the counterpart of the humans, it is been a long time hypothesized that nitrogen fixation occurs also in human gut, and indeed it is the fact, and many studies have demonstrated it. Nevertheless, these bacteria which fix nitrogen, and some have been well defined, like Klebsiella and Clostridiales, has been demonstrated to be part of these bacteria families which fix nitrogen. Even if this nitrogen activity does not substantially contribute to the host nitrogen balance, nevertheless, they are maybe similarly to what we have seen for Rhizopia in the plants. And if we look to other studies that says, and epidemiologically show that patients infected with Klebsiella, and particularly Klebsiella pneumoniae, and particularly those who have abscesses, pyogenic infection of the liver, they have a very high risk of colon carcinoma, which we know starts from polyps. So there is, just to tell you, myriads of hypotheses that we could bring together to understand that what is physiologically happening in the plant system that is being studied in agriculture to foster cultivation, new plants and something. We have to look at this previous experience to try to think in the same way for what is physiology and pathology in the human system. Because also in the gut, we have to understand the complex interactions and languages between host and microbes, and microbe interaction. Because as I said, we learned and we know that microbes work and stay together in a society. It's very difficult that the microbes makes pathogenicity or an action by itself alone. It's depending on other living creatures, so microbes particularly. And so we have to look at the systemic relations between microbes and bacteria. And of course, the type of metabolites which are part of the language and the symbiotic connection. Of course, also in humans, we have strong relation. Many studies have developed the important relation between the bacterial environment of the intestine and the brain function, and particularly also the liver. For instance, just to take an example of what is a very diffuse evidence nowadays in the world, that fatty liver and fat in the visceral tissues, the so-called adipose tissue, is influenced strongly by the microbiota, and alterations of the microbiota can induce the overgrowth of adipose tissue and induce also the inflammation that is an adding value for pathogenesis in this system. Steatosis, we know, is practically an epidemic that is spreading in the world, and the average 1 of 3 persons in the world have liver steatosis, which is actually like a red light flickering when we have a car and we drive. When the oil is going to fade, we have the red light. And this is the opposite in the liver, because we know that the fatty liver associate with a more higher risk of many diseases, and particularly those which are the major causes of death today: cardiovascular, metabolic, neoplastic, neurodegenerative, digestive diseases. But what is relevant is the fact that steatosis is like to have a kind of accelerator that need to be throttled by a foot to accelerate. And this acceleration is hepatitis in the liver or general inflammation in the body. So inflammation is a major factor working like a foot on the throttle of the accelerator to increase the speed to arrive to the problem, to the pathogenetical problem. And what is relevant is the important issue played by the microbiota dysregulation that can be triggering, the key triggering event in the inflammation, which can determine steatohepatitis in the liver and accelerate progression of all the diseases I have shown in the previous slide. So what variability of gut microbiota can depend upon is, of course, delivery is the first beginning of the development of the individual microbiota. Very relevant, for instance, the way we get to birth. Then from there, how we live in the early beginning to get the appropriate microbiota of our mother. That is very relevant. I will come back on this issue. Then after start, there are, of course, geographical, regional, of course, diet influences, host genetics, and of course, duration of life, age, and the different pathologies can impact. But also impacts mostly now in modern countries, particularly the iatrogenic influence of all the antibiotics used in the food and used also for medicine, and many other factors which are very relevant in impacting on the variability, and particularly also on the gradient that I was talking about before. It was very important. This gradient, the key issue of keeping this gradient is by the acid of the stomach. The key issue on keeping the gradient, that is the first step for having a good, normal, physiological microbiota, is the acid in the stomach. But if we don't care about this and we use without control drugs which inhibit secretion of acid, then we increase to have the so-called small intestinal bacterial overgrowth symptom. That is the increase of the number of bacteria and major region of fermentation, which is limited to the colon, increases and stepwise invade the ileum. So we have this tremendous epidemiological epidemic of SIBO that is mainly caused by the change of the microbiome, either in quantity and either in quality, because of the change of the quantity, as I said, in consequence of the overgrowth, and of course all the heterogeneity that is altered by the change in quantity. And then we have the overgrowth with consequence, the problem of the increased translocation of microbial products with an increased permeability of one of the major metabolic communicator that are lipopolysaccharides, that are inducers, potent inducers of inflammation. Lipopolysaccharides are major constituent of vaccines. What causes in vaccines the so-called hyperergic inflammatory reaction are adjuvants which are based on this type of substances. So one major issue is, for instance, the overuse of proton pump inhibitors, which actually make a kind of chemical gastrectomy. And this wide use for many decades has probably induced one of the major changes in the gut microbiota in modern societies. And this is clear. We do not need to go in the detail of what happens when we block the secretion of the acid in the stomach. And of course, a huge number of data have been appearing. In the beginning, the strong association was demonstrated. Then companies producing antacids and proton pump inhibitors started to boost the studies, making in a way that they did not show stronger correlation, only a little less. Because as you know, the results of a study depends on the methodology of designing it. So if you are smart enough, you can, in a statistical epidemiology study, condition the results from the design at the beginning. So coming to the issue of the methods to analyze microbes, we have to put questions. About microbes, which are the most important question? The most simple, naive question is who they are. Who they are these microbes? And we have the genetic definition of who they are by metagenomics, and then we make fantastic studies associating them to diseases and not to the diseases, making fantastic studies and good impact factor, but in practice, zero in knowledge, because transposing this in the human beings is like to understand if you are Italian, French, African, and then pretending to associate to disease an Italian or an African, or to say it is true, because if I say I am from Afghanistan, I can be associated with war now, of course. I am from England. No, I am not associated from war. But in terms of practical information, what is this information? Equivalent to zero. And in fact, all these studies have helped to identify which are genetically the bacteria in different places, but these associations are completely useless in clinical practice and also for identification of etiologies. Then we have the step to correlate the patient genome with the high risk. Is the similar study. The older history of man, since the ancient origin, the man has always been trying to understand if it is possible to predict the future. So prediction from Andromeda, from Aruspice, from Cassandra, and now we use genetics in the same poor way, even if we know that the genes represent less than 2% of the genome. The 98% of the genome is made by non-coding sequences. Sequences which help to use the gene. And since what actually happens in health and disease is not what you are, but what you do. Because you can have a potentially dangerous gene, but if you don't use it, there will be no damage. So this issue has to be considered consistently. And when we look at the microbiome, first of all, we have to understand when and where we are looking, even in terms of definition of which bacteria we have there, even only associating them with a certain status. So we have to understand where and time. And so it's very different according to where we look at. Of course, the most important step ahead in this preliminary naive way of associating microbes genetics to human genetics according to a disease or health status is at least to have in the single individual 2 time points and making a differential analysis of the microbiome pattern between 2 time points. This is already something, but 2 time points in the same individual. If you have 2 time points from different individuals, you start to jump on population study, and this is a very big garbage can, and you never get rid of some important information. What is really important is the changes in the same individual, at least from two time points. Of course, if instead of 2, you have 100 time points, dynamically, that's different. Another issue is, who are they? Okay, fine. What are they doing, and which other microbes? We know very well from easy studied area. The easy studied area are skin and oral mucosa, for instance, where it is possible to have very specific sampling. And from these studies, we know very well that bacteria live in biofilms, which have a dynamic growth from attachment, growth, maturation, the attachment, and there is a different behavior, a different synergistic work between bacteria, different jobs from different groups of bacteria. And what matters is actually the mature biofilm, which represent the healthy condition and the dynamic changing over time, but the stable condition is the mature biofilm in the dynamic life of the biofilm. And in fact, from the bacteria of the oral mucosa, a lot of studies have well demonstrated the relevance of the interplay between the different bacteria, the role of the bacteria for introducing the biofilm. Then the bacteria which make the so-called scaffold, if you wish, of the biofilm system, like the Fusobacterium nucleatum. And then in red, some bacteria which are critical in determining the triggering of inflammation, like parodontitis. Parodontitis is exactly one of the major reasons of systemic inflammation, which can work as throttle, as foot on the throttle of the accelerator of liver steatosis, or this metabolic condition of the organism. If you have, for instance, parodontitis, mostly you have steatohepatitis. Anybody who has parodontitis has steatohepatitis if he has a fatty liver. What we learn from this area, where, as I said, is very easy because we can get very easily the way and the type and the site where we can pick up the sampling. And here we see the importance of sampling. Oral microbiome in saliva is completely different than dental biofilm according to where we pick up. When we use the swab, the so-called swab, it depends where we put the tip of the swab and when and how we study dynamically the change of the dental biofilm. Completely different if you look in the garbage can of saliva, because saliva is the washing, is like the river that has crossed in the city of the mouth and taken out everything in a mix of everything. It's not actually detecting the biofilm and the group or family or working team of the bacteria in the biofilm in the gingival mucosa. Just this extrapolating this concept is just, we have seen in the pandemic how crazy and stupid, I would say, has been the world, sampling erroneously the trying to check the virus in the nose with few microliter in the tip of the swab. Instead, in this condition, the best would be to have a washing of saliva of the total mouth taken with a sponge that is autonomously taken, few minutes of chewing, then you have 2 to 3 ml of washing of all the oral cavity. And then you increase the sensitivity of SARS-CoV-2 detection, either antigen or molecular, over 100%. But the total incompetence of those who have sitting and talking or in committee, scientific committee, not having in the curriculum, never discover the virus, never establish a new technology for detection of antibody or antigen. So they are like Ronaldo speaking about boards on the America Cup sailing. And this is why we have started again today having a lot of negative sampling because of the stupid way of taking the sample. So saliva is very good for COVID, for SARS-CoV-2 detection, not good for detecting or studying the parodontitis bacteria which sit in the biofilm, just to making an example of which is the appropriate sampling according to different questions. Taking into account this example of SARS-CoV infection, bacteria are extremely important. Even in the mucosa of the respiratory system, we know we can actually inhibit, for instance, the entry of a virus like SARS-CoV-2 with bacteria, which inhibits the binding. This is just a very recent poster that was presented in an international meeting of microbes, just showing how important are potentially some probiotics, some lactobacilli and bifidobacteria. Some of these produces proteins on their surface or secrete proteins which block the binding of SARS-CoV to the epithelium and to the cells, SC2 receptor. So this is very interesting because, as we know, in these fields, there are a lot of industry which are looking for the future to use mixtures of bacteria to sterilize, for instance, the theaters, the surgical theaters, instead of having antibiotics or chemicals to kill bacteria, to keep the environment just using the natural way of keeping out pathogens, having the so-called good microbe environments. So coming back to the issue, the most important issue for study the relation of the microbiota with health and disease is sampling. And in sampling is critical also for the stools. Just if we classify the different stool we can have, and we have this way of taking samples, it's completely different if we have a stool type 1, type 2, type 3, or type 4, 5, and 7. If I have a type 1 or type 2, and I use the brush and the swab, I have to take the sample on the surface of this stool, and probably I will get some representative of the microfilm of the rectum and the last sigmoid tract of the colon. If I use this swab and I go inside the stool, I take the garbage can of the bacteria eliminated by all the gut. If I have diarrhea, I have lost everything. I just have the washing. So this is something, there is no work published, even in very famous journal, they never explain where they have taken, how they are taken. So, and then there is one sample of one person and thousands of persons believing to kill the variability with the number, completely wrong. We need to have also an implementation of knowing what they do. We know that the most important diagnostic assay for bacteria, let's take the example of Helicobacter pylori, comes from tests aimed to identify what they do. When you know the metabolic behavior of a bacterium, then you can detect the bacterium using a system to detect its activity. The urea breath test is the most specific assay to detect the presence of Helicobacter pylori in the stomach. the overgrowth or intolerance to lactose is another breath test. So the functional tests are very useful. So we have to understand that identifying what the bacteria are doing is also very relevant, and probably more relevant than just identify only the bacterium. One unmet need is, for instance, to develop, and there are some progress very important in this issue, to detect the activity of autobrewery, so the production of ethanol in the gut by bacteria, by either group of bacteria, because this production is not by a single yeast or bacterium, it's a community. So I forget about the community. If I have a functional test, I have the best for clinical practice, because I can have a functional breath test or whatever, where I have a substrate, carbohydrates, standardized, then I can measure in different parts of the intestine whether after this, in some time lapse, we can have the production of ethanol. So this is something that is very relevant, because it would really tell us how many of the so-called metabolic steatosis are really induced by autobrewery. Autobrewery, production of ethanol by microbes, can be impacting very strongly on the pathogenesis of steatohepatitis, as we know, determining, allowing lipopolysaccharide to alter pass to go to the liver and trigger inflammation. So I think in the complexity of the studies, metagenomics is relevant, but it is to be aimed not only to the genome of the host of the bacteria, but also to other aspects that I will briefly come to. Importantly is to combine them with metabolomics if we do not have a specific functional assay, because metabolic products can be the way to understand the language which is really taught at that time in that position by those bacteria. So it's a way to study what they do. And of course, it would be relevant to combine that with the immune response, because any bacterium whose product cross the mucosa of the gut are probably triggering an antibody response. And so this can be another part to be developed in combination with the other two. The other issue of genomics is that we have not to forget the most important genome study that we have to perform is the genome of the mitochondrial DNA, because bacteria, in their language with the body, talk with their counterpart, which are the ancestors of bacteria, mitochondria, that are being fixed in the cell. So mitochondria are ancestors bacteria, and they are communicating because they are the key for energy function. And this communication between gut bacteria and mitochondria is very relevant. The music comes from the play of the two. We know very well that one of the reasons that when we are born not naturally by cesarean cut, we can have trouble in developing a good microbiota relation with our genetics. Why? Because we inherited the mitochondria of our mother. We have all the mitochondria of our mother. And so the best for us would be to get the bacteria of our mother at that time, because those bacteria are particularly in harmony with if the mother is healthy, with the mitochondria of our mother. If we cut this beginning, you already make a very problematic start for the growth of the new symbiosis by the newborn and the microbiota. Many studies are coming up. Just point out a few of them already published 10 years ago, showing that the mitochondrial upper group are associated with different types of disease and lifestyles, and also risk of pathogenicity. And this is very relevant, because it just shows that the hypothesis is true, but we have many, many examples. There are different associations of haplogroups of the mitochondrial DNA associated with diabetes, with major diseases like the one we were talking before. So the studies between these relations and genome of the mitochondria will be much more important than the genomic study of the somatic cell. And the relation, of course, with the gut, the microbiota, because the mitochondrial production of reacting oxygen species plays a key role in the innate immune response, and of course can be a key role in triggering of inflammation. And the relation between mitochondria and bacteria of the gut is, of course, extremely important on this balance. And so we have to understand the regulatory mechanism which happens between these two populations, mitochondria and bacteria. So the microbiota targets host cell to ROS regulation, and so the participation of commensal bacteria in this type of relationship is crucial. And this is, of course, crucial for the very important point of triggering inflammation. As I said, is the key issue in the pathogenesis related with fatty liver and adiposity of the visceral tissue. And the microbiota modulate this probably in a crosstalk with mitochondria. So the study has to be combined at this level. And we know that in this then happens a lot of other issues. For instance, the so-called gene transfer changes which occur by plasmid within a bacteria to the DNA or mitochondria can be a critical issue for pathogenesis. And then in the future, maybe we'll understand that some diseases are etiologically induced by some of these agents that are transporting some gene from bacteria to cells in this way. So the integration of multi-omics meant as studies systematically of different features is very relevant, but add complexity to the issue. And we cannot believe that this complexity is solved by the number of cases. This is completely wrong view. We cannot imagine that we can get rid of the risk of the problem of the wrong sampling. And then it's not surprising that if you look at many studies on this issue, when we put together genomics, epigenomics, transitory, then the results are comics. In most of the issue, the results are at a little mouse out of a big mountain at most. This is the reason why we didn't have much developments in the last 10 years, in spite of a potent technological possibility. We need to understand that big data analysis is a tremendous power, but it is an instrument. And as in any statistics, we need to understand that one of the critical issues is the quality of the data that is influenced mostly by the quality of the sampling. And the quality of the sampling depends on the question we want to address in the study. According to the question, it changes the type of sampling, the number of sampling, the site of sampling, the way of sampling is not identical. This is something that unfortunately is not so taken in consideration currently, because currently we believe that science is technology. It's synonymous to, it's not at all. Science is art. Technology is handiwork. Worse than handiwork, because there is no possibility to correct. And artificial intelligence is an instrument, in the end, of human intelligence. most of the artificial intelligence applications are stupidity application because they are instruments badly used by a wrong concept trying to make an answer that was not appropriate for the good question. So the real point is to ask very well the question at the beginning. I stop here and eventually can develop some further concept during the discussion. Thank you very much.
Thank you very much, Ferruccio. I always admire your alternative, your somehow iconoclastic approach to the reality, the science reality. And I think that the issue you addressed today is a beautiful example of my understanding. So I open this fascinating and challenging speech to questions. Please, don't be shy. Is there any question from the audience? Raise your hands. Let me start.
May I intrude as an intruder?
Sure.
Just to say how pleased I am to see Ferruccio and to listen to him. And really astonishing the way he presents things. Though he has already decreed that I have a fat liver because I put sugar in my coffee, I think he is really one of our best testimonials in terms of what FIF can do. So thank you very much, Ferruccio, and I disappear. Thank you.
Oh, thank you very much, President. If I just can add a few things. Don't trust anyone in life. Just look at what they have done. My methodology that I understood to look for viruses was quite effective. Why? Simply because repeatedly successful. I was lucky enough to be part of the team who discovered the Delta virus, which is actually an ancient replicative RNA that is part of the transfer gene between plants, different type of living agents and human cells. It happens that I was a part of the team that discovered hepatitis C, and I was actually in the group of Michael Houghton, who actually discovered and was awarded of the Nobel Prize last year. Then in my own, we have discovered and defined the pathogenicity of hepatitis B E-defective virus, that is a mutant of hepatitis B, which causes the major reason of disease in the world, that is a subtype of chronic hepatitis B, the E-negative form. This is the condition why we have to say why I believe strongly in the what simply thing I have solved, because this is what I have learned trying to be successful repeatedly. When we were looking for Delta, at the time of PCR, nobody was talking about PCR. There was a blind detection in the gel on ethidium bromide staining. So we needed huge quantity of nucleic acid to detect the band in the gel, at least 10 to the 6 more concentration than now. At those times, we would not have been able to detect Delta RNA in the sampling if we would not have taken the right sample. I tell you simply, believing that this was a virus at the peak of the hepatitis and knowing that the virus at the peak is really killed by the inflammatory response, and we have a drop of the viral load at the beginning of the hepatitis event, I strongly advise a lot of patients that I was seeing at that time that were drug addicts at risk of being super infected with Delta, having a carriership of hepatitis B. I train a lot of them to tell me immediately when they could have some symptoms like feeling like I have a cold or I have influenza starting, a flu-like symptom, to call me immediately. One lady called me immediately one day in the afternoon. I took 300 ml of blood to have a lot of material. She had transaminases around 150. The morning later, she had 2,000 of transaminases. When we look for RNA in the evening, we got it. In the morning, we were negative because it was already fading down. At that time, without PCR, we would have lost the possibility to look at it. So the concept, I have taken this example to tell you the importance of the sampling when you have a scientific question. Very critical, much more important than the methodology to look at it.
Well, I agree with you, but there is one additional point in which, in my opinion, is very important. It's not only the possibility to have a good sampling, but also even more to have a functioning brain and then asking the right question. Because I think this is the crucial issue. So I fully agree with you that all the technologies we have are just tools to try to get an answer. But what we don't have to do is what a lot of researchers are doing now, and also the clinicians, to have the answer given by the machine without thinking. If you look at the patient and then you test the patient with 200 stupid and useless blood tests, then you're going to use just 2 or 3 blood tests to get the diagnosis. Otherwise, it's meaningless. And I think that your speech made clear that what is very important is, number one, to ask us and the rest of the team the right question, and number two, get the right sampling to get the answer, and then use all the tools you have at that time to ask. And I fully agree that the brainless use of artificial intelligence is meaningless.
No, but let's say it is important nowadays to realize that it's not possible anymore to have just everything in one person. We need to build teams. What is relevant is like in playing soccer. You need a good keeper, goalkeeper, a good defense, a good midfielder, a good goalie man. The point is that nowadays you need, for instance, if you want to address microbiota, you need an expert of microbes, probably an expert very good working in probiotics or working in the area of agriculture, in area where they have already developed well what we do not have yet in our area. So this is an important issue. The other issue, of course, you have a very good experts of artificial intelligence method and so on. So nowadays, the most difficult point is to build the right team. It's not possible, for instance, nowadays, when you want to have a good sample, you have to have also the good clinician, which understanding the question that you have posed, can think about the best way to get the good sample. On the other hand, it's not just one sampling with the life of a patient that is giving you information. This is something very, very relevant. So this is what is critical, because, Claudio, you know it well. The reason why you get money is not the beauty of the research, is how strong is your lobbyistic power.
Sure.
But after that, you need to have something very good inside. On the other hand, you have got the money, you have even a good position in the university, but you, in the end of your life, has made many zeros in the knowledge. But if you want to have a value inside the container, that is the point. So nowadays, the most difficult issue is really to build the right team according to the right question. So this is the point. And microbiota is a complicated issue because it's actually the top of complexity, because there we have to study the relation of a human system with microbes, with, as I said, not only the genome of the nucleus, but the genome of the mitochondria are relevant. And then timing and is relevant, and the way of course, of sampling is also very relevant. As you have said, the typical example is being even the simply condition of the pandemic has already made a lot of misunderstanding because of the sampling error, which were enormous in some condition, but not very relevant in other conditions. So this is and also technology is important, of course, because for instance, in the pandemic, we have not even standardized the technology. There is not been any work on standardization of the methodology to test antibody and the virus or the antigen. So the standardization of technology is very relevant and important. And of course, this fortunately is already present in many fields. For instance, nowadays, standardization of metagenomics or metabolomics is quite good. So I think we need really to approach in a systematic teamwork.
Yep, agree. Any other question, comment? If not, I thank you very much, Ferruccio, for this outstanding